Bismillahirrahmanirrahim. Assalamu alaikum everyone. Welcome to Stream of Knowledge. Today we will learn about gram-positive bacteria versus gram-negative. Gram staining is another criteria for classifying the bacteria into two categories, gram-positive and gram-negative. Basically, in gram staining there is a primary or basic dye called crystal violet is used. Gram-positive bacteria retain that dye while gram-negative does not. As they retain crystal violet dye, so they appear purple. Gram-positive bacteria appear purple, while gram-negative does not retain and they appear pink. We will study about gram staining in detail in our next video. You can remember the, the major difference between gram-positive and gram-negative by a mnemonic. For gram-positive, the mnemonic is PPT. Here, P stand for the positive and second P stand for the peptidoglycan. In gram positive, the peptidoglycan layer is very thick and teichoic acid is also present. While in gram negative, you can use the mnemonic long. L stand for the lipopolysaccharide, as we have studied in a previous video that lipopolysaccharide is outer membrane in gram negative bacteria and lipopolysaccharide is not present in gram positive. As you can see here, there is a peptidoglycan which is thick. We have learned that in gram-positive, peptidoglycan layer is thick and teichoic acid is present. As you can see here, there is teichoic acid and thick peptidoglycan layer. But there is also lipoteichoic acid. The major difference between teichoic acid and lipoteichoic acid is that teichoic acid protrudes from the peptidoglycan layer while the lipoteichoic acid is protruding from the cell membrane here like this. While in gram negative there is very thin peptidoglycan layer, but there is also a outer membrane layer which is absent in gram positive, that is lipopolysaccharide. One more special structure in gram negative are the periplasmic space. Between the outer membrane and the plasma membrane, there is a space called periplasmic space which contain enzymes, for example beta lactamase. This enzyme break down antibiotics, for example penicillin. That is why gram-negative are more resistant to the antibiotics. Porine proteins are also present in gram-negative bacteria that play a role in facilitating the passage of small hydrophilic molecules into the cell. You can also study the gram staining by this picture. Here is the teichoic acid which is only present in gram positive, and the thick peptidoglycan layer which is almost about 5 to 80 nanometer while only 2 nanometer in gram negative. And there is also a periplasmic space in gram negative bacteria. The major difference is the three components: lipopolysaccharide, which is also called endotoxin and only present in gram negative, while teichoic acid and think peptidoglycan, which is only present in gram positive. Cell wall is single layered in gram positive as it only contains peptidoglycan. As you can see here, there is only peptidoglycan. Here cell wall is only made up of peptidoglycan, while here cell wall is made up of not only peptidoglycan but also lipopolysaccharide. Hence, the cell wall in gram positive is single layer, while in gram negative there is a double layer. As we studied, the cell wall is thick as the peptidoglycan is thick in gram positive, while and is the gram negative thickness is 8 to 10 nanometers only. Peptidoglycan layer is thick, hair is thin. Teichoic acid present, teichoic acid is absent, lipopolysaccharide is absent, and here lipopolysaccharide is present. Lipopolysaccharide is also called endotoxin, which cause septic shock, while the outer membrane is not present. Here the outer membrane present. Lipid content is low, while the lipid content is high. There is the gram negative is resistant to antibiotics as the periplasmic spaces contain enzymes which break the antibiotics, while they are susceptible to antibiotics. As you can see here, the purple color of the gram stain, this is gram positive, while the pink color of gram staining means the gram negative. Thank you for watching. To subscribe, comment, share and like. Stay tuned for the next videos.